Hello, everyone. So today, 5 of us are going to present some perspectives on NEET Living Machines over the last 3 years in the program. So hello, my name is Anand. I'm a senior in majoring in computer science and molecular biology. Within NEET Living Machines, I majored in the immuno-engineering and computational biology tracks, and also broadly within NEET, I'm one of the student representatives.
Good afternoon, everybody. My name is Rachit Makamla. I am a senior at MIT majoring in biological engineering with a minor in computer science. And within the NEET Living Machines program, I was part of the immuno-engineering and computational biology tracks.
Hi everyone, I'm Grace. I'm also a senior in course 10B and 7, so chemical engineering and biology. Within NEET, I am part of the Biotech for Environmental Engineering program, and I'm also serve as a community building officer for the program.
Hi everyone, my name is Z. I'm a senior in biological engineering and biology, and in NEET Living Machines, I'm part of the immuno-engineering track, and I also serve as the mentorship liaison for the program.
Hello, everyone. My name is David Quabiado. I'm a senior majoring in 6-7, so computer science and molecular biology. I did the computational biology track, and I am the communications liaison as well as a student representative.
So at NEET Living Machines, students start off in their second year taking a class called 20051, which is basically an introduction to NEET Living Machines. This is a 6-unit class, and basically it introduces students to the 6 tracks within NEET Living Machines. Microfluidic engineering, immuno-engineering, environmental engineering, computational biology, immuno-engineering, and microfluidic device engineering. So the class, every week, there's weekly lectures that sort of cover each one of these tracks, specifically given by distinguished professors at MIT whose research aligns with these tracks, and they give broader perspective on the field. Also, within 20051, it's a very hands-on class. So the class focuses on applying the skills we learn in the classroom into microfluidics. So specifically, one of the projects is designing, modeling, and building our own microfluidic devices. First, we conceptualize the design using CAD software, we model it using computational software, and we build it in the lab. Another one of our cool excursions as part of the class is going to MIT Nano to fabricate our own devices, and there we learn techniques such as photolithography. And lastly, in project teams, we conceptualize our own organ-on-a-chip devices to sort of, for specific disease applications. We met with professors and grad students in labs across campus, and we sort of presented our final designs to the class. So basically, 20051 not only exposed us to the tracks, but also taught us about the design, build, learn, test cycle that's sort of paramount to biomedical research, and sort of allowed us to apply these skills to our own individual research projects in our desired tracks later on in our time in NEET Living Machines.
After 20051, NEET Living Machines students take 20054, Research Immersion, in which each student works in a research lab for over 1 year. And during their research project, they get a chance to apply all the skills that they've learned to 20051 and all of the classes they've taken through their NEET Living Machines track towards real-world research questions. So I was part of the immuno-engineering and computational biology tracks. So for that reason, I chose to join Professor Michael Birnbaum's lab in the MIT Department of Biological Engineering, where I had a chance to work on a project that tied these two fields together. So the Birnbaum lab is very interested in studying T cells, which are also my favorite cell type because T cells are arguably one of the most important immune cells in the immune system. They coordinate and direct responses against foreign pathogens that are infecting us. And they're able to do this because each T cell expresses a unique T cell receptor that is capable of binding and recognizing foreign antigens presented on the surfaces of cells. The Birnbaum lab is particularly interested in pairing these T cell receptors with their cognate antigen binding partner, because if we're able to understand which T cells, TCRs, bind to which antigens, we can understand what goes wrong in dysregulated immune states, such as autoimmune disease, where T cells recognize our own self-proteins and lead to unregulated inflammation. The challenge with this pairing problem is the vast combinatorial diversity of this particular problem. We, in each of us, contain billions of unique T cell receptor clones, and there's trillions of possible antigens. And adding to this problem, current approaches are extremely expensive, laborious, inefficient, and just do not scale enough to be able to capture the true incredible combinatorial diversity of this pairing problem. For my 20054 project, I aim to kind of tackle this issue by building a tool called T-Craft. T-Craft allows us to assemble and test over 30,000 T-cell receptor sequences in a single reaction mixture using modular combinatorial DNA assembly techniques. In order to build this tool, I use extensive computational optimization to design and optimize all of the components used in the reaction. And thanks to this computational optimization, which used a lot of stochastic optimization algorithms, we were able to demonstrate that every single component in the T-Craft reaction achieved an efficiency of greater than 99% when tested in vitro. Having built T-Craft, we could then use it to tackle very fascinating problems related to disease-relevant questions. So using T-Craft, I assembled 3,808 TCR sequences from patients who have vitiligo, an autoimmune skin disease. Thanks to T-Craft, we were able to assemble all of these TCR sequences in 1 week's worth of time in a single set of reaction mixtures at a price point that was 70-fold lower than existing approaches, thanks to the optimizations that were done. Having built all these TCR sequences, we could then go in and screen them against a panel of peptide antigens. And excitedly, we found a subset of TCRs shown on the left in the box, which were reactive against melanocyte and skin-associated peptides such as MART1 and PMEL, flagging them as potential promising immunotherapy targets for vitiligo. I'd now like to invite the other students to share their 20054 projects.
As Rack mentioned earlier, T cells are crucial to our immune system, which is why for my 20054 project, I work with Dr. Brandon Dukoski at the Reagan Institute, where we characterize anti-cancer natively paired TCR repertoires, meaning that we isolate cells from patients, characterize the natively TCR chains that are found in their body in an effort to create and understand immunotherapies that can be applied for these patients.
For my 20054 project, I worked with Professor Sangeeta Bhatia in the Koch Institute, and basically my project was delivering drugs locally to the tumor microenvironment using specialized nanoparticles. And basically throughout this project, we applied this drug delivery vehicle to different scenarios, for example, in lung cancer or in cystic fibrosis, where we wanted to do targeted genome editing, and also use machine learning in collaboration with Microsoft New England to sort of optimize these nanoparticles.
For my 20054 project, I was working with George Church at the Harvard Medical School, and it was about evaluating tRNAs, which are like helper biomolecules that help make proteins. And we were seeing how evaluating them on an evolutionary scale, so evaluating them across multiple species, could help us to develop our own tRNAs to create proteins with what they're called non-canonical amino acids. So not the base like 20 that you might learn in school.
Then for my 20054 project, as part of the biotech environmental engineering track, I joined Professor Ariel First Lab in the chemical engineering department, where I worked on engineering a microbe called Shionella onedensis. Essentially, this microbe can produce an electrical signal in response to various chemicals in its environment. And I worked on genetically engineering this to respond to certain chemicals, as well as perfecting the overall design of the reactor itself, which you can see on that side. So outside of our academic and research interests, NEET Living Machines has a really big emphasis on our community and our culture. So shown in the first photo is an image of the class of 2025 on our last day of 20051. Most of the people that you see standing in front of you right now are in that photo. And we also have a dedicated leadership team that meets regularly in order to determine the programming and the events that we plan for the rest of the community. So in addition to administering a lounge to provide a space for students to meet together and collaborate, we also have hold various events to really build this sense of community in our people. So for example, we hold regular dinner parties at Professor Linda Griffith's home. In addition, last year we began an initiative to host our own awards ceremony. We really seek to emphasize various seniors and other students who have really contributed to our culture. We also hold various academic talks to help our students understand the industry and the different research pathways. This year, we had a faculty dinner with Professor Ritu Rahman from the Mechanical Engineering Department. We also held in collaboration with MIT Biotech Group an AI and Biotech panel.
In addition to the events that Grace just mentioned, we also have a mentorship program that we run where we match students with mentors across academia and industry. So this program was actually launched prior to the class of 2025 joining the NEET program, but it was unfortunately paused during COVID. And last year we decided to bring it back, which is why in the spring of 24, we launched a pilot program where we matched 10 students with professionals from across industry and different areas, from companies like J&J, Novartis to Celino. And given the success of this program, we launched a FOVE program this past year. Here is a photo from this past fall where we had a kickoff mixer where industry professionals came in to talk with our students about their roles and as well to just offer some networking opportunities for our students. And this semester, we had an end of semester dinner where we had a roundtable discussion where the mentors were able to kind of like share their experiences with the program as well as for students to find their mentor matches. And last but not least, we also host a bunch of industry talks. Here's just a few events where I helped to organize, and over on the left is an event with Dr. Ebro from Novo Nordisk, where he brought in prototypes of the projects that he was working on, where we got to really see and go in depth into the different types of pathways that are available to us as students.
And yeah, with that, we'd like to say thank you so much for listening. And just from a student perspective, I think all of the students here can agree that this has been a really transformative, really wonderful program to be a part of. I've really personally have appreciated all the mentorship I've gotten through, all the industry talks, all the networks and connections, the wonderful letters of rec that Mehdi has written me that's gotten me scholarships. I appreciate it all. And I would say this has been a really transformative experience, this program. So I would like to say thank you.